All right, so students who are online, do you have any questions regarding how the session is going to be going? You can either unmute yourself or you can talk in chat.
Answer the question. The correct option will be displayed when we click again.
Okay.
Okay.
All right, so we'll just start. So who wants to answer the first question? You can raise your hand. No one's raising their hand? All right, so a bunch of them have answered it's B. They're right.
Then the C.
All right, here's question number 2. Who wants to answer this one? So everyone's answering C. And you're right. Here's question number 3.
ما بحضرها.
All right, so this one, a lot of them seem confused. What do y'all think? Okay. So A is the correct answer.
Okay, so before we move on to the next question, since we have, for some questions, we have different questions, and some people keep going, so maybe we should discuss for the students online. If they have any doubts.
Oh, so they should, like, they will explain why they picked the answer? Okay, so who wants to explain? May come closer to the mic. I can't come closer to the mic if you just open it. My mask is going to help. Am I more audible now? Oh, Dr. Muhabat. No, so the other. Yeah, okay. So Dr. Muhabat asked someone to explain the option. So for question number 3, who wants to explain why the answer is A? I pick Mariam Zaitoon. Mariam, you can unmute yourself and explain why you picked the answer to be A.
Yes, good morning, doctor.
Good morning.
I picked A because I found that the others are wrong. So, for example, E, I remember it was the cyst that was contaminating the food and water. For D, it is 4 nuclei that are for the Entamoeba histolytica, not 8. And C, they don't have flagella. B, there was no animal beverage as the main reservoir. So that's why.
Good. What about E? Why is it not E?
I said it was the cyst, not the trophozoite that was ingested.
Good. All right. Now we'll go back to the answer. So for question number 2, Ziad, if you can explain why you give the answer to be C.
Okay, doctor, because the guardian ambia is GI tract and it infects the GI, while the Entamoeba invades the tissues. The Naegleria foliaria also invades the tissues. The Trichomatis vaginalis also invades the tissues. And the Plasmodium vivax invades the blood mainly.
Okay, good. All right, now for question number 1. The answer is B. Al-Walid Hijazi, if you want to explain why the answer is B.
ضل يطلع لي فتوى الكرتي ما مش راضي يتغير اسمي هنا.
Al-Walid Hijazi. All right, Labiba, if you can explain why the answer is B. Or why you and not C.
Yeah, all of them has a direct life cycle, and the human is the definitive host. But Onchocerca volvulus has indirect life cycle, where I think the definitive host is the black fly or the intermediate.
Good.
Yeah.
Good. All right. That's it, right? So what's the definitive host of Onchocerca volvulus? Good. All right. Next questions. Okay. Who wants to answer this?
Nobody's going to say it, so just pick someone.
All right. Who do I pick? You. What do you think is the answer? The girl wearing pink. The girl wearing pink. Yes. You don't know. Okay. Who knows?
It's an exercise, so don't
You don't
try.
necessarily have to be picked. All right. So some people online are answering. Marwan, why is it B? Students here. Okay. Okay. All right.
It's B, doctor.
They can try, but I mean, in the interest of fairness.
All right. Yeah.
I think it's B. Do we need to explain one?
Yeah.
Okay, so first of all, we need to know that what I can see is that it's an acquirinated egg. So we left Parasitology vaginalis because the Comonos vaginalis only found in the Protozoic form. And the rest are passed in the vestibular form. And here the Lambda is famous for this structure because it's the effective structure here.
Okay.
Yeah, so what stage is this?
This is the effective stage.
Yes, I know, but what is it?
This is
This is friendship.
So what Dr. Muhabat just said is that this is the cyst stage of Giardia lamblia.
I told you that how it's for Giardia lamblia.
Good. Everyone who answered B, good. Next. So who wants to answer this one? Someone can decide. No? Okay. All right. Why is it A? Good. So it's Tina Solium.
Yeah, but why in B wrong?
Because in this
The larvae.
the larvae that will not go to
Sorry.
Because
Because I
what?
can see people
I should tell
online.
them. Yeah, yeah, yeah.
I think I saw something else. Maybe, yeah. I don't know.
I don't know why it's not me because what I know is that Tanya showed you what it does is that it once it's inside, let's say, becomes the cirrhosis inside the egg, and then once it's infected, the egg won't that's what causes cysticercosis inside humans.
Okay.
So as the undercooked meat pigs.
Okay. So when you said it forms the satire in the pig, when humans ingest that form, what happens to the parasite from humans after ingestion in the flesh of the pig? So what happens? What happens to the larva? It becomes the adult form, right? In the GI, right? Okay. But if the egg of Peninsula are injected, what happens to the egg in the human home? So why do you now think B is the right option?
Because the adult form is found in the skeletal muscle, and now the skeletal muscle use plants. So if we eat this undercooked pig meat, we can also get infected with the eggs and not only
No, the larva is the juvenile one, so it's not mature, it's not fully. It's only the adult forms that will develop and then we have immature, mature, and gradually produce. The eggs are released from the granulosus, right? So when the larva form is ingested, it develops into the adult form. However, when the eggs are ingested, they develop into larva. They don't become adult. Because humans are not the usual intermediate host. They are supposed to be the definitive host. And remember, I told you during the lecture that Victinia saginata, Victinia solium, both the eggs and the larva are infected. However, Victinia saginata is only the larval form that is infected. Okay? Is it clear now? You're sure? You're convinced that D is not the right answer?
how can where are the eggs released? Like, through our stool, the contaminated water through their stool. Okay, I think that's good.
Any other question?
Doctor, can you explain it again for those who are online?
Are those online in those?
Yes.
Okay, so what I said is that with Tinea solium, both the eggs and the larva form are infective. When the larva forms are ingested, they develop into adult worms, so they cause GI infection. However, when the eggs are ingested, humans now act as accidental intermediate host. We are not we are the definitive host, not the intermediate host. So the eggs will develop into larva, not into adult form. So the larva can form in any organ of the human body. That's why we have it as cysticercosis. Is that clear now?
Good morning, doctor.
Yes, good morning.
I think the correct answer is C, because as you said, the humans are the definitive host, so they produce the embryonated eggs, and thus the feces of an infected human should be the answer.
Drinking water contaminated with feces of an infected human. Oh, I think you're right. I think you're right, yes. Yes, the right answer is C. I agree with you. Because the pigs are not the definitive host, right? The adult worms are found in humans. I agree with you. Should correct this.
She's still there. Bushra is still so the doctor explained, but
why is it not? D. D will cause GI infection. Okay? D will not lead to ingestion of eggs. When you have undercooked pork, you have the immature larva in the flesh of the pig. Okay? Are you following, Bushra? So that cannot give you eggs. Remember, it's the eggs that will develop into the cysticerci in the different organs of the body. So D cannot give you that. D will give you intestinal infection. Are you clear now? Any other comment? Okay.
next question. So who wants to answer this one? Whenever you're done reading. Does someone have an answer yet?
Yes.
Okay. Why do you think it's B? Alright.
Why are they wrong?
So I think you have to come near the mic and explain to your colleagues. Because they have suggested A, C, and E. So you should explain why A, C, and E are not answers. Yes, the answer is B. Rima.
Okay, so I think it's not A because it's indirect life cycle. Okay, what is the mic?
They can hear you.
Okay, I think it's not A because it's indirect life cycle. And for C, it's not the longest, it's very short. It's only three proglottid. And for D, because the embryonated egg, I think, is the infective form, not the larvae. No, sorry, because the larvae does not go to skeletal muscle. Usually they go to the brain and the liver when they are ingested. And for E, the main one transmission. Yes, it's ingestion of eggs from the feces of the dog.
But the reason why it is not the right option is because they don't cause abscesses.
Okay.
The doctor said the reason why D is not the answer is because they don't cause abscesses. Does anyone have a question? Does everyone understand this question's answer? In person, everyone understands? Online, everyone understands? Dr. Do they form high data sets? Yes, Iman. Alright.
Yes, so I've just discussed the last slide.
And then after this is a normal lecture?
No, there's no lecture today. It's just interactive session. Yeah. Thank you so much. Okay. Thank you. So I received a question from one of you, and the student stated that he still doesn't know how to differentiate between Babesia and Plasmodium under the microscope. So he wants me to elaborate more on it. So here we have two. What type of film preparation is this? Thick or thin? Thin. Why do you say it's thin? The red cells are preserved. So here you have a blood film from a patient with Plasmodium falciparum infection. And below it, you have the blood film from a patient with Babesia infection. As you can see, the trophozoites in the two films are difficult to differentiate morphologically. So I told you that unless you are really looking out for it, you may miss it. Most often than not, in malaria endemic areas, when these are seen, they are reported as Plasmodium. So it's difficult to differentiate the trophozoite just on morphology alone. For a trained microscopist, there are certain things, particularly when you are in an area endemic for Babesia, you can see extracellular parasitic forms. Somebody who is not so trained in reading slides for Babesia will miss all these forms and just report it as Plasmodium. So when you see just trophozoite, it's very difficult. However, if you see the merozoites have a particular arrangement referred to as the Maltese cross. So you find them as 4, even when they are more than 4, they tend to have this orientation, which is quite different from what you find with Plasmodium. As a result, the Maltese cross, when you see the merozoite, is what can help you to make a diagnosis. You cannot, unless you are well trained and you are looking out for it, you can hardly make the difference between the trophozoites based on morphology alone. Is that clear? So to the person who asked this question, I hope it's clearer now. If you're online, let me know if you still have areas of doubt. Okay. Oh, no. Is it up? Right. Yes, Babesia have Maltese cross merozoites. Well, it's the arrangement. It's not as if the merozoites becomes Maltese cross. It's the arrangement that we refer to as a Maltese cross. Okay, Bushra? Any other comment? Babesia has Maltese cross merozoites. Okay. Yeah. Any other comments for students online? So that's it. Any comments from students in the class? So that's it. You have an idea of the way the questions will come in the exam. So basically, the questions are going to be on life cycle, infective, diagnostic form, and things like that. Appropriate samples to send, laboratory features, and things like that. Okay? So have a good day. Any more comments from students? Okay. Okay. So we're done.
Doctor, can you please explain it again?
Which one?
The answer, because I couldn't understand it.
Which answer?
Between how can we differentiate Plasmodium and Babesia under a microscope?
That's what I said. You can't differentiate the trophozoites because they are so similar, right? So you can only, if you get the merozoites, that can help you because the merozoites have a peculiar arrangement, which we refer to as the Maltese cross. So if you get the trophozoites, unless you've been practicing in areas that is endemic for Babesia and you've been seeing a lot, and you now start identifying these extracellular forms, that is what can guide you. But as medical students, I don't expect you to, know this. Even experienced microscopists at times mix things up. Okay? So when you see the trophozoites only, that is not enough. If you find a sample, only trophozoites, no merozoites, you may have to perform molecular assays to be sure. Okay? So looking at trophozoites only is not enough. Is the first one Babesia as well? Exactly. No, it is not. This is Plasmodium. Okay? Sameha, this is Plasmodium. The second one is Babesia. That is why I looked for, slides that are so typical of the two conditions. And the second one, and the other two are Babesia, right? Yes. These are the trophozoites of Babesia. And this is showing the merozoites with the Maltese cross arrangement. Any other queries? Do we need to know every step of the life cycle? Or is knowing infective and diagnostic stages enough? I've had this question before. I don't understand how you can recall infective and diagnostic form without recalling the life cycle. How? One moves on to the other, right? I don't know how you can do it. So if it's convenient for you to recall the infective and diagnostic form without recalling the life cycle, that's fine with me. If it's okay with you, if that's the way you can learn I don't know. But left to me, I don't see how you can know the infective and diagnostic stages. No, I'm not going to ask you questions like that. If I'm going to put any SAQ questions, they'll probably be on prevention and control. And that I believe you must know how the infection is acquired for you to be able to prevent and control it. Yeah. Exactly. So how do you now remove the life cycle from the discussion? Every step, I like to remember the overview of the life cycle. That's why you have those diagrams. Those diagrams are a summary of a lot of things. That's why I'm not going into the biology of the parasites. So you have a man, an arrow is going into his mouth, and then you have a diagram that shows something. Or you have an insect, the portion where the insect is feeding on is blown up. That's telling you something. So I don't know, I honestly don't understand why you keep asking, do we need to know the life cycle? I don't get it. Because the life cycle is the core of the discussion, if you ask me. It's only telling you the interplay between the infective and the diagnostic stages. So if you want to remove the life cycle from the discussion and you're comfortable with that, it's okay with me. Okay? Will we be asked to identify the parasite just by their eggs? How do you think I should answer? Okay, as a medical doctor, when you send a patient sample to the lab, what do you want the lab to do for you? Iman, can you please answer the question? please, if you send the patient sample to the lab, okay, what do you want the lab to do for you? Iman, go ahead and mute yourself. I meant as an SQ, you will have to identify different points, characteristics. Do you think you can explain the question to me? I honestly don't understand what you mean.
So I think what she means is, so for example, she'll be given like an image. Iman, is this what you mean? Like you get an image of the, like for example, the slide here, and then you're asked to identify, like, mention 3 points why this is a parasite of, yeah. So just characteristics, just listen to characteristics. And of course you're studying the characteristics anyways, aren't you?
Okay. So if I give you eggs of Taenia species, and I ask you, what is the life cycle of this parasite or something like that? Is that a fair question? It's not. It's not a fair question. What do you think? That's a fair question, right? Say something. You're all looking at me. This is not fair. It's supposed to be an interactive session. I'm the only one doing the talking. You know what interactive means? It's a two-way discussion, right? No, that's okay. Iman, that's okay. I can read your comments. So just, yeah. Continue to send in your comments. What, you think that's not a fair question? Why? Because I said no life cycle, did I?
If you're not studying the life cycle, what are you studying for micro?
No.
Yes, but you have to know the important points of it. So for example, in a life cycle, you end up with parts that are not really related to your patient at all. And so forgetting those might make sense. But there are relevant things like the doctors mentioned, the infective and diagnostic stages. It's part of the life cycle, honey. That's how that works.
Like, sorry, I didn't hear you.
Writing down the steps. Oh, so you mean like as an SAQ? She means like as an SAQ, like write the life cycle of Giardia lambda, and then you
Who
have to
does
write.
that?
Like,
you're not zoology students.
yes, yeah. So that's separate. Yeah. So it is the doubt that everyone.
The biology of the parasite. So in terms of discussing the life cycle as it should be discussed, we didn't go into that. I only gave you an overview of how humans get infected and how the infection is perpetrated in the community. So we talk of the infective form, then the diagnostic form. The infective form, how does it get into the environment? Or how is it picked up by a vector if it is a parasite that has a vector, okay? So those are the key points. So when you say, do we need to know the life cycle? It's a little bit confusing to me because the interplay, like I said earlier, between the infective and the diagnostic form is what is depicted in the life cycle. So if you have a patient whose sample turns out positive for a particular parasite, naturally it should come to your mind the likelihood of acquisition of infection, right? Then you have to make up your mind, is it protozoan helminth, because you have to treat. And you may need different medications to treat, right? So when you say, do we need to know the diagnostic form or something like that? My question is, as a doctor, you saw the patient, you sent the sample to the lab. What did you expect the lab to tell you? I believe at the end of the day, you want to treat that patient. So there must be evidence of infection before you start your therapy. Your sample, or rather the patient sample which is sent to the lab, will show certain forms of the parasite that the lab will now report back to you. So the natural question is, what forms of this parasite did the laboratory see? So you should have an idea. Exactly. So that's why we went through the whole idea of discussing cysts.
Doctor, I think what everyone's confused about, it seems, is what kind of SAQs are going to come.
I don't know. That question should go to Dr. Artis. Okay? I don't know. I have no idea. If I do, I will have shared it with you. I can assure you. So that question should go to Dr. Artis. Okay? Yes, Iman, you're right. Those are the key points of every life cycle of every parasite, okay? You should lay emphasis on the infective and diagnostic stages, histology, their route of acquisition, and what area of the body is affected. Okay, so any other questions? Yes.
Ask you, when are we going to take the second lecture?
From?
Under.
I told you there's no second lecture. We're having just an introduction. Yeah, that's why we converted it to this discussion session. So it's just discussion, and then once we're done, we're done. Okay? No, no. Any other question? So, yes.
For the content, there was a biological classification.
Somebody has asked me that question this morning. Do you need to memorize? I don't like the word memorize, personally. With microbiology, if you memorize, you may run into trouble. I can assure you in the exam, everything is going to the names can be confusing. Rather, you should learn. Okay, you can have mnemonics to help you, but try not to memorize. That's it. Do you need to know Ascomycota, blah, blah, blah? Yeah. Yes, you need to know the names, but you don't need to go into details. You notice that I didn't go into details. If you check my previous slides, I did go into details of each group. But Dr. Artis said, make it simple. Okay? So things have been changed for your own group. So you should know just the basic thing. Ascomycota. Let me tell you something. As medical students, when you interact with your friends who are not in medical school, say some things to confuse them. That's the beauty of being a medical student. I mean, if I'm having a discussion with my friend who is not a medical doctor and I say, Ascomycota, she's going to be like, speak English. So I said, okay, don't worry. Those are fungi that have their sexual spores in a sap. That's all you need to know. And that makes the discussion interesting. Your friend is going to be wowed. Yes, I can assure you, unless you didn't say it with confidence. Say it with confidence. See, as medical students, what you are learning now are common things that happen in the environment. As medical students, you can create awareness. You just need the little basic detail. Okay? Don't be scared. These names were, put up by men, human beings. Okay? So it's not something from the space. It's not something from the moon or from Mars. Okay? And keep it at the back of your mind. I'm sure the very first lecture you had with Dr. Anna, she must have told you the reason why we name organisms and classify them. We name organisms so that when we discuss, wherever you are, we are on the same page. So if you tell me that you isolated Staph aureus from a patient sample in Riyadh, a doctor who is practicing in China knows what Staph aureus is. You understand? So you're all on the same page. So if you say this Staph aureus is resistant to methicillin, the doctor already knows what that means. So it's just to unify the discussion and to also differentiate different agents from one another. So Staph aureus is different from Klebsiella. If you gram stain, you're going to see different things. So that's the whole essence. So don't be scared. This is going to be the tool of your profession. You're going to be mentioning the names of these organisms forever, unless you don't practice. So this is the time to learn and, impress your friends, let them know that you are in medical school and you are learning something different from what they know. That's the beauty, okay? So please don't be afraid. Yes, you can go ahead. Sarah, you can ask your question.
Assalamu alaikum, doctor.
Wa alaikum salam.
I wanted to ask about the plasmodium for the schizont. I don't exactly get what it is. Like, I know you described it as a coin in a purse, but I don't get it. Is it the actual cell acting as the schizont, or is it something within the cell?
What do you mean? When the merozoites that are released from the hepatocyte at the end of the exoelectrocytic cycle invade red blood cells, they start to develop, they form trophozoites. The trophozoites will mature further, and within the red cell, they form the structure referred to as the schizont. Now, the schizont itself contains many merozoites. So that's why I refer to the structure schizont as the bag of coins, or the purse filled with coins. When the schizont breaks down, merozoites are released. These merozoites will go ahead to infect new red blood cells, and that perpetuates the electrocytic cycle. Now, why does the organism develop through these different stages? I don't know. That's what we know about the parasite. Why it decides to act that way, it's something peculiar to that particular parasite, okay? One thing I should also state is that when you start seeing schizonts in peripheral blood, that is a very severe infection. Usually the schizonts are formed deep down in tissue, okay? Because I told you the organism is microaerophilic, so it tends to actively divide in tissue. Then the merozoites are released. The merozoites you can find in peripheral blood. Is that clear?
Yes, thank you.
You're welcome. Any other question? Okay. So have a nice day, and I wish you brilliant success in your exams. Okay.